Let's Talk About Pain, a podcast from the Quebec Pain Research Network. In each episode, we discuss the latest advances in pain research with guests from across Quebec in the health and research communities. Enjoy your listening.
Hi, my name is Daniel Ocay, and I'm a PhD graduate from McGill University and now a postdoctoral research fellow at Boston Children's Hospital. Today, I'm going to tell you about personalized treatment for chronic pain. Proof that it's possible, advances in research on the subject, and are we closer than we think? A biopsychosocial approach to understanding pain is known as the most successful model to date, such that it encompasses the interactions between biological, psychological, and social components specific to each individual. In other words, it's essential to tailor treatment to the individual patient. Since two patients with the same diagnosis differ physically, socially, and psychologically, lumping them together in the same treatment program is unlikely to produce the best results when compared to personalized treatment. With a biopsychosocial approach to pain management, chronic pain patients could improve their physical and psychological conditions such as depression and anxiety and enable them to return to their normal life activities. During the World Pain Congress 2022, held in Toronto, I was fortunate to attend several thematic sessions touching on the subject of personalized treatment. One of the sessions I attended was on the use of sensory testing to profile chronic pain patients. This is a technique I used for my PhD project. Sensory testing is a non-invasive tool for evaluating the peripheral nervous system, such as the place where someone reports their pain, and the central nervous system, which is at the level of the spinal cord and the brain. Different tests are available, which include touch, pressure, vibration, heat, and cold. Studies show that sensory testing is useful for profiling patients for treatment. However, it is not clear which tests are best suited for profiling patients. I was fortunate enough to interview Dr. Jan Vollert on this topic. Dr. Jan Vollert is a bioinformatician and professor at the Imperial College London. His research focuses on the application of statistical models in pain research, mainly quantitative sensory testing of patients with neuropathic pain. He is also a member of the International Association for the Study of Pain and the Neuropathic Pain Special Interest Group. Here are snippets from our interview. So I'm here with Jan Vollert. So we're really just talking about, personalized treatment itself, and there has been a lot of research regarding sensory testing and how it can be integrated within personalized treatment. So how do you see QST can be used in clinical settings to stratify patients? Do we see this coming in the near future? Is it being already applied in clinics to, help patients towards more personalized treatment?
Yeah, I think the question is how do we define QST in that case? If we take in the wider definition, then I would say yes, it is used now because then it includes bedside sensory testing protocols, and I think they are being applied and employed now, and there's some really great protocols being developed, particularly in Germany, that's based on the German QST protocol, but also in Boston around Roy Freeman and colleagues.
So you say it's pretty much used in some clinical settings itself, and how have patients received that? Is there any nerves in terms of these tests itself? Because sometimes there may be a little bit of pain inflicted. How, for example, the German population accepted QST within their clinics or the clinicians themselves?
QST does have a burden for the patients, and we shouldn't ignore that. The more complex protocols, comprehensive protocols, just take time to begin with. And you need to be really focused on it, particularly if you met the thresholds. You need to be on time. That does take a burden, and that is relevant to patients. So in that way, I think it's important that we go to protocols as simple as possible while still being just as feasible.
When you mean simple, you're talking a lot about bedside sensory testing, if that's correct? Yeah. Do you still see any obstacles when it comes to personalized pain treatment regarding QST itself?
Lots of them. Lots of them in the sense that there's so many unanswered questions that we're still working on. Lots of what we're doing, we're just starting to employ in like wider populations, wider clinical populations. And there's so many questions that we always had that we could have never answered before. And we're just going for it.
As you just heard from that interview, Dr. Vollert says personalized pain treatment is possible. However, there are still barriers that we need to overcome. I also had the chance to interview Dr. Martine Bordeleau on this subject. Dr. Bordeleau is a postdoctoral researcher at the Université de Sherbrooke in Quebec. One of her main areas of interest is improving sensory evaluation methods by involving the research community. Here is what we discussed. I asked Dr. Bordeleau if whether we would be seeing more sensory testing in clinics in the near future. She responded that before we can see sensory testing in the clinics, there is still lots of development and research needed, particularly in establishing more normal reference values, which takes into account the ethnicity of the individuals. Currently, there's not enough consideration on this subject, even if multiple articles report that race and ethnicity may impact the results of the sensory tests. Moreover, the medication intake of the patient, the sex of the person conducting the tests, and other sources of variability need to be considered. In the current database of reference values, we are more representative of a Caucasian population. Therefore, there is work to be done to have reference values representative of our diverse population. Dr. Bordeleau also emphasized that there is work to be done on standardizing of our procedures. She says that although sensory tests give us a quantitative reportable number, this number needs to be confirmed by complementary approaches, such as simply discussing with the patient to ensure that the number we record and can indicate any anomalies in sensory function actually is related to the patient's abnormal sensory function. This can be done by discussing about the different sensations with the patient during the sensory tests, but this is not fully integrated in the protocols. Dr. Bordeleau wishes that sensory testing moves away from strictly being quantitative but qualitative as well, which means including the patient in the sensory tests to acquire the full picture. Although Dr. Bordeleau has been working on this since 2018, to integrate change sometimes requires good timing, but a small spark may be all it takes to finally see this change come into fruition. Another interesting session I was able to attend was on the use of pain biomarkers as a basis for decision-making in the therapeutic development process and in clinical practice. A biomarker is any measurement that indicates a state or condition of an individual. For example, inflammatory molecules in our blood can serve as a biomarker for arthritis. In this session, moderated by Dr. Mary-Anne Pellimalter, who has over 25 years of experience in scientific research and over 20 years experience in drug discovery and development, researchers described their progress and lessons learned in developing clinical biomarkers to profile patients and a clinical biomarker signature to facilitate treatment decisions in patients whose chronic pain has a severe impact on their daily life. Dr. Pellimalter began the session by pointing out that biomarkers can significantly improve the probability of a successful therapeutic development. But which biomarkers should we be looking at? That depends on what we'd like the biomarker to do. Firstly, Dr. Tony Blockhill, a research fellow in the School of Physiology, Pharmacology, and Neuroscience at the University of Bristol, discussed biomarkers that target the nociceptive system, which is the process by which we feel pain. This approach was chosen because nociception can be divided into several stages. Many of these biomarkers have been studied in animal and human models to assess the effects of drugs. Dr. Blockhill has demonstrated that changes in brain recordings in animals and humans are noticeable after the administration of certain drugs, and therefore can be a potential biomarker for the future. Secondly, Dr. Katie Vincent, a women's pain researcher in the Department of Obstetrics and Gynecology at the University of Oxford, discussed the profiling of patients with chronic pelvic pain, which is common, affecting around 24% of women globally. Diagnosis of the pathology is difficult and frequently requires several surgical interventions, often under different specialists. So it's perhaps not surprising that biomarker research in this field has focused on non-invasive diagnostic tests. There are over 100 biomarkers for endometriosis, but none has been shown to be useful in clinical practice just yet. Dr. Vincent's project takes a different approach, aiming to use biomarkers to identify pain drivers rather than the presence of the disease. Dr. Vincent presented a multi-center study that integrates detailed phenotypic data with plasma genomics and proteomics and metabolomics in a cohort of nearly 800 women, 556 with pelvic pain and 230 pain-free controls, and extends this dataset with additional questionnaires and psychophysical assessments in a subset of them. In addition, their design allows them to specifically explore associations of endometriosis and bladder symptoms. Dr. Vincent presented the results of integrative analyses of this rich dataset and demonstrated that it's more complex than we think. Biomarker profiles can help us better understand pain in specific groups. Ultimately, Dr. Vincent hopes that this strategy will identify new therapeutic targets, inform a more personalized approach to treatment, and refine preclinical models to optimize drug discovery. Finally, Dr. Laura Simmons, professor of anesthesiology, perioperative medicine, and pediatric pain at Stanford University, discussed an ongoing project entitled Signatures for Pain Recovery in Teens, or SPRINT for short, which integrates 4 parameters: blood, which could include immune markers, sensory testing, imaging, which looks at brain structure and function, and patient-reported measures, which includes demographics, physical, and psychological factors. These selected parameters have been shown to be associated with pain and physical function, but were limited by the sample size of study participants, single-parameter assessments, and moderate effects. This study represents the largest cohort of adolescents with characterized musculoskeletal pain to derive a prognostic biological signature of pain and functional recovery by exploiting a multivariate computer analysis pipeline to extract reliable results from this complex dataset. So what about personalized treatment for chronic pain? Is it possible? Short answer would be yes. Several studies show that each individual is unique in their experience with pain, and a biopsychosocial approach to pain management is the most successful model for enabling a return to normal life activities. Are we closer than we think? Yes and no. Certain tools or biomarkers are already being used in specific pain clinics, but there are still challenges and work to be done to improve personalized treatment. I hope you enjoyed today's episode, and I look forward to talking to you more next time. Until next time, thank you.
Thank you for listening to Let's Talk About Pain. Don't hesitate to subscribe so you don't miss an episode and to share the podcast with your friends. For more information, visit our website at qprn.ca or our YouTube channel by searching Quebec Pain Research Network. See you next time.